going to start the recording. Okay. So this review session is going to be over exam 2 and all of those important topics that you really need to pay attention to before actually attempting the exam. So let's dive right in. So first up, we're going to talk about passive and active transport because those are still relevant, right? As we saw in cellular respiration, where we're cheating at active transport when we get to the electron transport chain, which of course we'll talk about later. But right now, I just want to cover the basics. So who can tell me what passive transport is, and does it require any ATP? And you can respond using your microphone or you can send it in the chat. Good. Okay, so I'm seeing diffusion and no ATP is required. Very good. So with passive transport, we're going with the concentration gradient. Right. So we're going from a high concentration to a low concentration. And because we're going with the concentration gradient, no ATP is required. And an example of that is diffusion. We also learned 2 other examples, like facilitated diffusion, which is what you see right here when you have those protein channels and the carrier proteins. And then we also learned about osmosis. That's another type of passive transport. Now, what about active transport? So what's the difference? Do I need ATP in active transport? Right, I do. And why do I need ATP? What makes it different from passive? Right, so we're going against the concentration gradient. So we're typically going from an area of low concentration to an area of high concentration. And that's what we're seeing right here. Good. So are there any questions generally about passive versus active transport? You just kind of need to know the definitions and which one ATP is involved in. Are we good? Perfect. Thank you. So now we're going to actually dive in to the 2 main chapters that we went over so far, that being metabolism and cellular respiration. So first, what I want to talk about are the laws related to metabolism. The 2 primary ones being the first law of thermodynamics and the second law of thermodynamics. So who can tell me what the first law of thermodynamics says? Because these are super important. And to help you out, remember these are the laws about energy. Very good. Okay, so yes, the first law of thermodynamics says that energy cannot be destroyed nor created. So it's just transferred or transformed. So maybe we get energy from the environment. And if you look in your PowerPoint, there's like a picture where there's energy being transferred from the sun to a plant. And then maybe an animal comes by, like a rabbit, and eats the plant, and that energy is then transferred to the rabbit. So it's not coming from just anywhere. It's just circulating from the environment to animals and then back to the environment. Good. And then I see what you said, Isaac, about mass cannot be created or destroyed. That's actually the law of conservation of mass. But so like first law of thermodynamics and law of conservation of mass kind of parallel one another, except the thermodynamics law applies to energy and the conservation of mass, of course, applies to matter. Good. Okay. So then who can tell me what the second law of thermodynamics says? Remember, this one is also about energy. Entropy, right? So with the second law of thermodynamics, we have incomplete transfers of energy. So some is lost as heat to the environment, And when we talked about this in class, we went over a specific picture of like a bear eating a fish. And so when the bear eats the fish is a form of energy, right? The energy is being transferred. That's the first law of thermodynamics. And then the second law of thermodynamics, so when the bear actually does activity, some of that heat, some of that energy is lost as heat to the environment. So that's kind of how the two are connected. Good. So you just need to know the definitions, basically. What do those laws say? Because those are super important when we're talking about metabolism. So are there any questions about that? You guys did a really good job explaining those. Okay, so if there aren't any questions about the laws, that's kind of the introductory stuff to metabolism. Then we're going to hit catabolic versus anabolic pathways because this is the bulk of the metabolism that we talked about, especially with catabolic. As we see when we move into cellular respiration, which is an example of a catabolic pathway. So, who can tell me the general definition of a catabolic reaction or a catabolic pathway? What are we doing? Okay, we're releasing energy. And what do we need in order to release energy? What's the precursor to that? Are we breaking things down? Are we building things? Good. Yeah, so a catabolic pathway is basically going to break a molecule down, and the result of breaking those bonds releases energy. So that's where it comes from. Good. So how does that differ from an anabolic pathway? What are we doing in that one? It requires energy, right? Good. So anabolic is different in that we're building molecules, and in order to build molecules, we have to input energy. Yes, Rebecca, I like that example too. So when Dr. Annette talked about catabolic reactions in class, she talked about the first part of catabolic, which is kind of cat, and how cats are destructive little creatures and like to push things off tables and break them. You can also think of it that way. Thank you for bringing that up. I think that's actually a good way to remember that too. Okay, so those are the general definitions, and those will 100% come up on the exam because they're very important, and we talked about them a lot. So make sure you're clear on what's happening in each pathway. Okay, so we're good on definitions? Okay, perfect. So then next, we're going to talk about the diagrams. So what I've been telling people is that for the metabolism section, in order to really study for the exam and be good on all the material, you need to know how to do the in-class activity we did for metabolism for both catabolic and anabolic pathways. So the one we did in class was for the catabolic. So we drew the free energy diagram, we spontaneous, non-spontaneous, exergonic, endergonic, positive or negative delta G, that sort of thing. So if you can do that for both catabolic and anabolic pathways, then you should be perfect for this exam as far as metabolism goes. That's how I would recommend studying for it. But we'll go over the free energy diagrams really quick. So if we look at the one on the left, is this a catabolic or an anabolic reaction, and why? Good. Catabolic because we're releasing or we're losing energy. And are catabolic reactions endergonic or exergonic? Yes. So catabolic reactions are exergonic. And remember, energy exits, exergonic. You can make that connection too. So in an exergonic reaction then, do we have a positive delta G or a negative delta G? Good. Yes. And is it a spontaneous or a non-spontaneous reaction when we don't have the extra speed bump? Good. Yeah, so it's a spontaneous reaction without the speed bump. So then I want to ask, is this reaction that's modeled on the left, is it spontaneous or non-spontaneous with the way that it's written? Okay, so it's actually non-spontaneous, right? Because we talked about if there's an extra speed bump, and I know this says spontaneous, and exergonic reactions in general are typically spontaneous. That's why it says it couples the two. But if I take a look at my graph specifically, I have this little extra speed bump. Can anyone tell me what this speed bump is called? It has a specific name. Activation energy, very good. And the activation energy is basically the energy we need to put in to get the reaction started. So if I'm able to overcome my activation energy, does my reaction become spontaneous then? I'm able to get to the top of this hill. Yes, very good. So it's just about overcoming that activation energy. And how can I do that? How can I overcome the activation energy for a catabolic reaction? How can I get over this hill? Catalyst. Yes, good. So a catalyst lowers the activation energy and it allows the reaction to go faster. So what's an example of a catalyst that I could use? Enzyme. Yes, enzyme, a protein or favorite biomolecule. Yeah, so we can use an enzyme, which is a catalyst, to lower the activation energy. Very good. Okay. And then, of course, on the graph, we have our progress of reaction, or time, on the x-axis. And on the y, we have our free energy, or delta G. Good. Okay. So that was basically the in-class activity for catabolic reactions. So let's do it for anabolic. So how do we know that this graph is anabolic? Besides just like process of elimination, we've already done catabolic, so naturally this one has to be. Reactants are lower? Yeah, okay, good. So when we have an anabolic reaction, we said that we require an energy input. So naturally our products are going to be higher. Good. So is it endergonic or exergonic? Ender. Yes, it's endergonic. So remember, energy enters and then ender, they kind of sound the same. So that's how you can remember that. Is it spontaneous or non-spontaneous? Good. Yeah, so it's non-spontaneous. If we put our little person right here and we pretend they're going to slide, they're not just going to magically go uphill. So if we think about it that way too. Okay. And I think that's pretty much it for all of that stuff that you need to know. So are there any questions about the free energy diagrams, endergonic, exergonic, spontaneous, non-spontaneous? Oh, is it a positive or a negative delta G? That's the last question I want to ask. Positive. Good. Good. Okay, now are there any questions about it? Or is everybody feeling pretty good with the free energy diagrams? Because I know we've spent a lot of time in class going over these and making sure everybody's on the same page. Are these the diagrams? So these are not the exact diagrams, but you will need to know how to look at a diagram that's given to you. So something of this nature, And if she asks you if it's catabolic or anabolic, you need to know how to identify that. So you would look at where the reactants are and then where the products are, and if energy is being released or put into the system, that sort of thing. And then she may ask you like about how to identify where the delta G is. And it's always the difference between your reactants and products. She could also ask you how to identify the activation energy, and you would have to know that's the extra speed bump right here. No, you will not be making a graph, but she'll probably give you a graph, and then let's say this is identify part A, so it would be this, it would be delta G, and then your answer bank may be A, activation energy, B, delta G, C, reactants and products, something of that nature. Does that make sense? Good. Okay, so it'll just be something like that. And I just saw a few people came in, so just for the sake of getting accurate attendance, I'm going to take a quick pick. Okay, so let's move on. Okay, so we just went through this, but just to do it again for the people who just came in. Catabolic reaction, is it endergonic or exergonic? Good. Is it a positive or negative delta G? Good. Spontaneous or non-spontaneous in the general sense? So without the speed bumps. Good. And is a catalyst ever needed for a catabolic reaction? And why would one be needed? So, okay, yeah. So if we have a speed bump, if we happen to have a speed bump, that is when we would need a catalyst because we have activation energy. And in order to overcome our activation energy, we would need a catalyst in that scenario. If we don't have an activation energy, then that's when we wouldn't need one. Good. And we already went over the anabolic reactions, and everybody's feeling pretty good. What did you say the space represented? Oh, right here. So this is just the delta G, right? So the difference in the energy between the reactants and the products, that's delta G. Okay, everyone's favorite thing to study, cellular respiration. So what I tell people to study for this one is the in-class activity that we just did. So if you haven't turned that in, please turn it in because I'm going to start looking at those with Jasmine and we're going to grade them and we're going to post probably a solution so that everybody can double check their answers and make sure everything's 100% right because the in-class assignment is a really good study tool for the cellular respiration activity. And basically, this is what I outlined. So for each of these steps, you need to know, like where is it happening in the cell? Does it require oxygen? Like, is it a part of aerobic respiration? Which we know if it's aerobic respiration, it requires oxygen. And then if it's anaerobic respiration, that's when you don't have oxygen present. And when we talked about anaerobic respiration, which was very short, a section in our slides, but that was about the fermentation and like the buildup of lactic acid in the muscles. Because when we get pyruvate, we convert it to lactic acid and then that builds up because it doesn't have anywhere else to go. We need to know like what are the reactants, like what are we starting with in each step. We need to know what we're ending with in each step. And then, why is this step important? So we're just going to go through each of the steps of cellular respiration, and you are going to tell me what happens at each step. Okay, so starting with glycolysis. So where does glycolysis happen in the cell? Cytoplasm. Good. And does it require any oxygen? No. Right. So this step takes place in aerobic respiration and anaerobic respiration. It takes place in both. So we don't need oxygen. So what's my starting molecule? Glucose. Yes, glucose is a 6-carbon sugar. And when I get to the very end of glycolysis, I break down my glucose into what? 2-pyruvate. And I have a question. Why does it take place in both? So the reason why glycolysis can take place in both aerobic and anaerobic respiration is because oxygen is not involved. So for aerobic, if oxygen is not involved at that point, it's not really going to matter until you move into the mitochondria, which that takes place in aerobic respiration. But for anaerobic, you don't have any oxygen. So that's why glycolysis can take place there too. So it's not oxygen dependent. Does that make sense? Okay, good. So yes, I see 2-pyruvate. Yeah, and the 2 is important too. We get 2-pyruvate or 2-pyruvic acid. It's the same thing. Okay, what else do we get out of the deal, or is 2-pyruvate all that comes out of glycolysis? 2 ATP. Good. Yeah, we see this right here. And do we get anything else, or is it just ATP and pyruvate? NADH. Yes, we get 2 NADH. And who can tell me what NADH is? Like, what's the significance of NADH? Coupon. Yes, our coupons are electron carriers, and we see those become really important when we get over here to the electron transport chain. Good. Okay. So that's glycolysis. Another thing I wanted to talk about with glycolysis is that there's an energy investment phase and there's an energy payoff phase. So who can tell me what the energy investment phase means? Energy comes in. Using energy. Okay. Yes, you invest 2 ATP. So remember, in order to get glycolysis started, we initially have to put in those 2 ATP. We have to put in some energy to get it started. That's the energy investment phase. And then the payoff phase comes when we're actually going through glycolysis and we end up making 4 ATP total. But when we're looking at a net total, so overall, we only really gain 2 ATP out of the deal because even though we made 4, we used up 2 to start the process. So remember that. That's also super important with glycolysis. Okay, so that's the first step. Our second step is the prep step, right? And where does the prep step happen in the cell? Good. So we're moving into the mitochondria. We're taking our pyruvate. We're going into the mitochondria. And what comes out of our prep step? Acetyl-CoA. Yeah, and we get 2, right? Because we have 2 pyruvate. Each pyruvate is going to go into the prep step, so that means we do it twice, and we get 2 acetyl-CoA out of the deal. What else do we get? Do we get any ATP? No. Good. So we don't get any ATP. Do we get any electron carriers, like NADH, for example? Yes, we do. We get 2 of them. Very good. We get 2 total as a result of going through the process twice. Which part of the mitochondria does it occur in? So this is kind of like it's just coming into the mitochondria, so it's like at the very edges almost, if that makes sense. But you don't need to know like specifics, you just need to know that it's in the mitochondria. Good. Okay. So we got our NADH, we don't get any ATP out of the deal, which is a bummer, but whatever, we'll make some more later. And we get acetyl-CoA, which is important for the next step. Does the prep step require oxygen? It does. So anything in the mitochondria requires oxygen. Because remember, in aerobic respiration, we do glycolysis and then we enter into the mitochondria for all of the rest of these steps. In anaerobic respiration, we just do glycolysis and stay out here in the cytoplasm. We don't enter the mitochondria because we don't have oxygen, so we can't do all this stuff. Does that make sense to everybody? So if we're in the mitochondria, we need oxygen, 100%. Okay, good. So we go from our prep step, which is our second step, to the citric acid cycle. So who can tell me what's going on in the citric acid cycle? So acetyl-CoA combines with something else to form something. Okay. Sorry, I was trying to get some more people on the list. Oxaloacetate, yes. So our acetyl-CoA, one of them is going to go through the cycle first. It's going to combine with oxaloacetate to form what? Going to combine citrate, right? Which is also called citric acid. They're the same thing. And then we're just going to basically break down citrate to form another oxaloacetate, which is going to combine with our other acetyl-CoA to go back through the cycle. Because remember, a cycle is just like a process that loops. So that's exactly what's happening. So what are we getting out of the deal here? So we know that we make some ATP, right? Get 2 ATP. So we have the same yield as glycolysis, but what else are we getting here that's really important? Yes, we're getting 2 FADH2 and 6 NADH. So 8 of our coupons for our electron carriers total. And we know that those are really important, right? We're getting ready to see why. Okay, and does this step require oxygen? Yes, it does. Good. Okay, so electron transport chain. That's our next step. Well, our next step collectively, you don't really have to know this as much, but it's called oxidative phosphorylation, and it includes the electron transport chain as well as the ATP synthase and the chemoosmosis that happens with that. So oxidative phosphorylation is like the umbrella term for that. Yes. So our acetyl-CoA going to enter the citric acid cycle. It's going to, because remember the CoA is like the little flag that basically allows it to be recognized by the citric acid cycle. It's going to combine with oxaloacetate to form citrate or citric acid, right? Citric acid cycle. And we're basically going to break the citric acid down and then form the oxaloacetate again, which is going to combine with our other acetyl-CoA and do that same process over. Does that make sense? Good. Okay. So electron transport chain and ATP synthase, so oxidative phosphorylation. That's what I was saying. So got this graphic. I really like this graphic, and I sent it out to everybody. I think on sometime this week, I think. And it shows you pretty much everything that she drew on the slide. If you weren't able to really keep up with that, this kind of has everything, and it's color-coded. Very nice, very easy to follow. So we can just go through this. So electron transport chain. So in our electron transport chain, and all this is happening in the mitochondrial matrix, right? So we're like basically looking at the crestays, so like the folds, like the countertops of the mitochondria. And what are our electron carriers doing here? Like, what's their purpose? What are they doing that gets the process started? Smuggling, right? So they're donating those electrons, and those electrons basically help these proteins that are a part of the electron transport chain smuggle these protons or pump the protons into the inner membrane space. So we're cheating at active transport. So we're going from a low concentration down here and we're pumping up into a high concentration. And remember, active transport normally requires ATP, so the electrons are basically allowing us to cheat at that. Okay, so that's basically what's happening with the electron carriers and the electron transport chain. Does that make sense to everybody? So we're just pumping protons into the inner membrane space. Okay, good. So then the next part of oxidative phosphorylation is when the protons or the hydrogen ions flow back down their concentration gradient and they pass through the ATP synthase protein. And the energy generated from this flowing back down its concentration gradient and going through this protein basically powers the ability to make a bunch of ATP. Okay. Where did the protons come from? Okay. So if you look right here, the NADH, and do you see where it becomes NAD plus? And you have a proton. That's because a proton, if you look at a hydrogen, it has one proton, one electron. So when it donates the electron, it basically has a positive charge and becomes H plus. Does that make sense? Okay, good. So that's where the protons are coming from. They're coming from your electron carriers, and then they're pumped into the inner membrane space. And then when they flow back down their concentration gradient, they diffuse. So we're going from high concentration to low concentration. That basically, how Dr. Annette describes it, it like powers like the turbine almost, kind of powers it up and gets it ready to produce a bunch of ATP. And how much ATP can we see from this process? 32 to 34, so about 30, yeah. And the reason why you see so much variability in those numbers is because it differs from time to time, and it also differs between like prokaryotes and eukaryotes. So that's why you see a bunch of variation, but if you know that it's about 30, then you should be good. Okay, and of course we have this right here. So I know this was a source of confusion for a lot of people. Like, where does the water come from? So some of these hydrogen ions are going to be left behind, and they're going to combine with the electrons. So we're getting rid of the extra negative charge, because remember, we talked about how that extra negative charge just floating around in tissue is bad. So it's going to combine with our hydrogens. So a plus charge and a minus charge, those are going to cancel each other out. So we get H2. And then our oxygen is going to combine with the H2 to make H2O, right? And where does this oxygen come from? What's the relationship there? Normally, she would do a big gasp right about now. And it scares me every single time because I'm usually working on something else. But it's molecular oxygen, right? When we breathe in. So this is the connection that we see between ventilation, breathing, inhaling, exhaling, and respiration, which is when we break down molecules to produce ATP for the cell. Does that make sense to everybody? Good. Okay. So, if that's all that I have, this is probably my shortest session. So you're welcome. Does anyone have any questions or anything else want to go over? Tomorrow we're probably going to start going over mitosis, and I'm not 100% sure about how far we'll get into it, but I know that you all have had some like prior exposure in lab, so hopefully that won't be too bad. But are there any questions? You sent another study guide like last test. So actually there is one uploaded if you go to the current eLearn page, I think. And I'll send out like a screenshot or like where to find that exactly because I'm not 100% sure myself, but I know it's up there. And yes, so there will be a Kahoot as well. And I'll get Dr. Annette to upload that because we talked about, me and Jasmine went through the different Kahoots we have and we selected one that we feel would best like represent what's going to be on the test. So we'll definitely get that up there too. So you all can use that as a study tool. Okay. Any other questions, comments, or concerns? Remember about your research lessons. So make sure you've passed research lesson 3 and research lesson 4. Those are super important because you're going to use both your article and your website to write your paper. And the paper is due like mid-November, I think. And you can always check those dates by going to our eLearn page, and there's like a Making Connections essay tab on the side, and that has all of the resources that you need to write your paper. And you also have to, get a writing tutor to review it, so make sure you're keeping up with all of those dates as well. And of course, make sure to turn in your in-class activities so I can give you a grade and we can give you a great study tool. Okay, thank you, Jessica. So the final paper is due November 13th. Okay. Anything else anybody wants to add? I'm part of the attendance. Okay, let me make sure I got everybody before anyone leaves. This is just for my data. Yeah, I've got everybody now. Everybody should be good and should be on the list. So thank you for coming. I hope this was helpful to you. And if you have any additional questions, you can send me an email at any time and I'll try my best to get back to you as soon as possible. But thank you all, and I hope you have a good night.